Okay, our next speaker is Charlie Song from the Waxman Lab at Cincinnati Children's. He'll be speaking about HDAC1, retinoic acid, signaling, and the second heart field.
So good afternoon. So I would first thank the organizer for giving me the opportunity to present my work about HDAC1 requirement in second half development. Our lab is starting the congenital heart disease because, as you know, this is the most common birth defect. About 1% of all newborns suffer from these diseases. And about 1/3 of a congenital heart disease are partially, at least, attributed to the outflow tract defect. So it will be important for us to understand the mechanisms about how outflow tract develops. Like all vertebrates, the heart of zebrafish is composed of two separate populations, the first heart field and second heart field. As you see, in the light blue indicates the first heart field already differentiating cells, and the dark blue is second heart field that differentiate later and add to the first heart field derived from permanent heart tube and contribute to the ventricle and the outflow tract. So to explore the mechanism of how the second half develops, we start our project from a new identified mutant named cardiac repressive, for short for CRAG, from Alex Net-Product's lab. As you can see from this figure, the CRAG mutant in 48 hours have multiple developmental defects, including the reduction of pigmentation, the curved body shape, the initial edema, and yolk tension. But more important for us, when we look at the heart becomes more linear and shorter. In addition, we count the number of cardiomyocytes, and we found the number of ventricular cardiomyocytes becomes smaller, but not atrial. So it indicates the CRAG mutant has a specific heart development defect. To address when this defect first occurs, we went back to first heart specification differentiation. And by looking at the specific marker GAL4 and HAT2, at soma stage, we did not see any significant expression pattern between the wild type and the CRAG. And the same applies to MAL7 at 20 soma stage, a differential marker. So it suggests the defect is not somewhere from the early stages. So then we went to the late stages, 36 and 48 hours, and look at those cardiomyocyte marker expression. As you can see, the reduction of the cardiomyocyte expression at 40 hours become more evident in the CRAG mutant. And in particular, when we look at LTBP3, which is a signal-specific marker, it has been shown reduced at both stages. So it strongly suggests that the CRAG embryos have a specific second-half defect. So of course, we want to know what mutations cause this CRAG phenotype. So we did the positional cloning and found there is a mutation near the gene HDAC1, histone deacetylase 1. And further analysis by RNA-seq specify this mutation is a T2A expansion donor of exon 7 of HDAC1, in which they have the transcript either with exon skipping or intramutation. So moreover, we look at the protein expression of HDAC1 by Western blot, and you can see the HDAC1 protein is not detected. So the CRAG is a nonsense HDAC1 mutant. This is a little surprising for us because we know HDAC is expressed in many places and it has only a specific defect in the heart. So we like to explore the underlying mechanism of how HDAC is required for second half development. Early studies in our lab revealed the CYP26 deficient embryos have a similar phenotype as we saw in the CRAG embryos, which have a reduced ventricle in both the morphology and the number of ventricular cardiomyocytes. CYP26 is a family enzyme responsible for degradation of retinoic acid. And when CYP26 is gone, the retinoic acid goes up. A classic model of RA-mediated gene regulation is the R receptor plays a role as a transcriptional repressor with other co-repressors bound to it. And when RA binds to the R receptor, it becomes activator, and those co-repressors are replaced by co-activators. Since we saw a similar phenotype between the crack and the CYP26 deficient embryos. So we proposed in both cases that there are ectopic gene expression through the RA-dependent pathway. So we would like to identify what genes are ectopically expressed in both deficient embryos. So we look at the gene expression profile by RAR and HDAC in both deficient embryos. We included 3 groups of embryos for its analysis, including the crack mutants, the broad HDAC inhibitor-treated embryos, which share a similar phenotype as crack, as well as CYP26 A1C1 morphin. And we found only 58 genes upregulated significantly in all 3 groups. Among them, there one gene will be intrigues us, named the Ripply3. As you can see, it is upregulated in both the crack and CYP26 deficient embryos. Especially when you look at the isolated heart of CYP26 deficient embryos, it has been more upregulated. So it suggests that Ripply3 plays a specific role in heart development. So the other reasons about having Ripply3 as a strong candidate as an effector of HDAC1 is Ripply3 has been first identified gene highly associated with Down syndrome. As you know, it has a high probability of a congenital heart disease. And the Ripply proteins also have been shown playing a role as T-box protein co-repressors. And when Ripply3 is gone, there are ventricular septal defects. And more importantly, when you look at expression of Ripply3, it has been shown in the pharyngeal ectoderm and endoderm, but not the mesoderm. So we would like to ask whether the Ripply3 ectopic expression in the heart, in the mesoderm, would lead to the defect we saw in the crack embryos. So to address this question, we first ask whether the R receptor in HDAC1 actually represses Ripply3 expression. So we predicted 2 R response elements in 5 kb upstream of Ripply3, and we found the two sites has been enriched in R receptor ectopic expressed embryos by cheap qPCR. And since we know HDAC play a transcriptional repressor through the histone modifications, we would like to know whether the histone modification becomes changed when the HDAC1 is lost in the 2 sites. So we look at the H3K27 acylation, which is an active histone mark. When the HDAC1 is gone, it is enriched. For the repressive histone mark, H3K27 trimethylation. When HDAC is gone, it can be less enriched. So this study suggests RA receptor and HDAC1 both have been potentially shown as a repressor of Ripply3. And since we identify Ripply3 as a potential effector of HDAC1, we would like to see whether overexpression of Ripply3 can induce a similar phenotype as we saw in the crack embryos. And we inject the mRNA of Ripply3 and look at the heart in 48 hours. And we found the heart becomes linear as what we saw in the crack mutant. And similarly, we did the cell counting for the cardiomyocyte, and we found there a reduction of the ventricular cardiomyocyte by NAS atrium, which share a similar trend as what we saw in crack embryos. So, in summary, this is the first study showing the requirement of HDAC1 specifically for the second heart failure development. And we proposed a model in which the wild type, the HDAC1 R receptor maintains the histone modification in a repressive condition. And in turn, no Ripply3 is expressed in the mesoderm, and the second heart failure develops normally. And when we have an ectopic RA or the R receptor HDAC1 get reduced, then the histone modification becomes more active, and then the Ripply3 gets expressed in the mesoderm. And in turn, somehow through the TBX Ripply3 interactions, reduce the second half field. So this is a novel mechanism which I think has been really explored in the second half field regulation. So finally, I would acknowledge the people who involved in this project. So I'm happy to take questions. Thanks.
So I was trying to remember what Ripply3 was and why I'd heard of it. This is in the Down syndrome critical region, right?
Yeah, it has been first identified. That name actually is critical region.
So I know that you're talking about ectopic expression in a different tissue, but do you think overexpression of Ripply3 in other tissues could also be causing issues?
Yeah, actually, since we have not done the conditional induction of the Ripply3, so we don't know. But now I have a Zebrafish raising up which can be induced. The Ripply3 can be reduced conditionally and at any development stages. So that can help us to answer this question. Actually, the Ripply3 has been really explored in the heart. So I think there is possibility it can play a role in the tissues surrounding the heart. Yeah.
Nice work.
Yeah, so if you're thinking there's a general second heart field defect, you also see changes in the outflow tract or maybe in the aortic arch arteries in the HDAC1 mutant?
Since our lab is mainly focused on the heart, so we have not looked at those. But since we propose the defect in the actually second half of progenitors, so I would expect you to see the defect in all cell lineages derived from the second half of progenitors, so including the outer.
I think you would too.
Charlie, I was wondering, why do you think that the second heart field is so particularly affected? I mean, in the case of HDAC, you could potentially reason maybe there's maternal supply that is sparing the first heart field, but in the case of your overexpression of Ripply3, I wonder why it's so specific seemingly to the second heart field.
So what we think is, in terms of the HDAC1, which is expressed in many places, so the HDAC, since it's a co-repressor, it has to be recruited by other transcription factors, in this case like RA receptor. So those transcription factors can be differentially expressed in different organ systems. And another thing is, since HDAC plays a role in histone modifications, therefore we think the histone modifications before those regulation of other transcriptional factors, they might not be the same in the first place. So that might also make the different tissue responding to the same factor differently.